Hi, welcome everybody. We may as well get a start on. It's very exciting today to present to you the next generation of PhD students from the Centre of Animal Science. These students, three of them are presenting at the Beef 2021 conference in Rockhampton in May, and the other three are also attending and working at the QAAFI booth, which we will be having also at Beef 2021. So it's really exciting to have the students share with you today their sort of 5-minute presentation about their thesis. Three of them, as I said, the first three are presenting to the producer audience. So they prepared their talk with that sort of flavor to be producer audience, so that you're aware of that. But any scientific questions today is totally fine. So first, my name is Ala Tabor, as it's on the slide, and halfway through the presentation, I'll transfer the facilitation over to Professor Mary Fletcher. So halfway through, Mary will introduce the second three, I'll introduce the first three. So Acknowledgement of Country, it's the next slide. I would like to acknowledge the traditional owners and their custodianship of the lands on which we meet today. On behalf of the traditional owners, we pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Just a few more housekeeping things to mention. The seminar will go for about 45 to 60 minutes and should be completed no longer than by 1 p.m. Each presenter will speak for 5 minutes and there's time for questions. If you could pop them into the Q&A box at the bottom of the screen, that would be great. And there'll also be time to ask questions after all the talks have concluded today. So don't use the chat for the questions, use the Q&A. And if you are asking questions at the end, let us know to who the question is directed if it's after the six have already finished. So our first speaker today is Harry Lamb. And Harry is just in his beginning of his second year of his PhD. And he's supervised by Professor Ben Hayes and Dr. Liz Ross. And he's investigating the ability for real-time DNA sequencing to occur in the crush to increase the speed and accuracy of genomic selection in Australia's northern beef industry. Harry is also currently the QAAFI Student Association President, and if there's time at the end, he'll speak to that as well. So, welcome, Harry.
Thanks very much, Ala. DNA is the building block of life. We've all probably heard that one before. But what exactly does that mean? And most importantly, why do we care? A, C, G, and T, the four building blocks of life. Four tiny Lego pieces that make each one of us who we are. Each one of us on this Zoom call have 3 billion of these building blocks crammed into every one of the almost 15 trillion cells in our body. We each share 99.99% of these 3 billion building blocks with the person next to us. But it is the 0.01% of variation, or what we call mutations, that make some of us taller, give some of us blue eyes, or unfortunately for the blokes listening, make some of us go bald. These mutations can be small, one Lego block in size, which we call SNPs, or they can be large, hundreds to thousands of Lego blocks, inserted, deleted, or rearranged. These we call structural variants. Cattle, like humans, also have 3 billion building blocks in every cell, and it is the same small percent of variation between animals in a herd that makes some cattle poll, some cattle more fertile, or other cattle cranky. Thanks to research over the last 20 years, we now know which mutations are responsible for over 20 different traits. For example, we know that animals with a structural variant on chromosome 1 will be poll, or that animals with a specific SNP on chromosome 14 will be more fertile. Knowing the different mutations an animal has is called a genotype. And until recently, genotyping was only possible in a lab. In 2013, a small portable DNA sequencer called a MinION was released. A DNA sequencer is a device that reads DNA like a VCR reads a cassette. The sequencer, which you can see here is about the size of an iPhone, can read DNA anywhere, even in the cattle yard. All you need is purified DNA to load into the device, and you get the sequence of 3 billion Lego blocks that make that animal in front of you. Imagine you could calculate genomic breeding values for your calves while you tag and brand them. You take a tail hair sample when they enter the race, and by the time the calf is in the crush, you know every genotype you need to predict traits such as pole status, temperament, potential number of calves, mature weight, or intramuscular fat content. This might sound too good to be true, but with the help of my supervisors, I'm working on a pipeline that would see this become a reality. Crush-side genotyping, as we like to call it, will allow producers in the northern beef industry to generate genomic estimated breeding values on farm within minutes. This would allow even the most remote properties to use genetic information to select desirable animals from birth, rather than waiting until an animal has matured. This reduces generation interval and rapidly increases the rate of genetic gain in a herd. Crush-side genotyping is certainly not a reality yet, but the genomics field is moving faster than ever, and COVID has seen the development of this technology accelerate. In our lab, we've already sequenced almost 100 animals and are working our way through this data to develop an accurate, user-friendly pipeline for predictions. In the next 2 years, our goal is to be able to produce accurate genomic breeding values in under 20 minutes per animal in the lab, and then trial this pipeline remotely on farm. We believe within this time frame, we could realistically see a couple of hundred animals genotyped overnight, ready for drafting in the morning. From there, our challenge will be putting this into an app so you can generate predictions at the click of a button. Technology is worthless if people can't use it. So our goal is to ensure that one day you, your stock agent, or your station hand can walk over to the cattle yards with a min-iron in your hand, ready to draft. Thank you.
Thanks, Harry. That was great. So don't mute or put your camera off, I'm sure. See if there's any questions. I guess the question from me is, how would you see this physically working? Like, would there be like a training program? How can you envisage this happening in the future, if that makes sense?
So one of the ways we see this rolling out initially, I guess, would be have consultants visit farms. So this would be sort of replace the role of a stock agent, and then they would help with that. But then eventually, really the goal would be to actually make it user-friendly enough that producers can go ahead and do it themselves. That would just completely remove the need to actually get external people out to the properties to do it for them.
That sounds good. So there was a question about, someone just said they can't see slides running. We're just letting you know there is just one slide per presentation. Jeffrey Fordyce is asking if it's worthwhile discriminating the differences between genotyping and genomics. That's for you, Harry.
That's quite a good question. I guess keeping a industry audience in focus, I think discriminating between the difference may not be necessary. I guess really trying to emphasize that genotyping, we're looking at just the variation in a herd. We don't need to look at the whole entirety of the genomics data, and really would wrap it into a complete pipeline so that no data analysis would really be necessary by a producer.
Okay, I'll just have a quick look. There's no more questions. Someone's asked a question a bit about the format of this seminar. This is the format of how the students will be presenting at beef, hence this is why we're doing it this way. And we invited 3 other students who are also attending beef but not presenting at the seminar. So I'd like to thank Harry for his presentation. There's opportunity to ask questions at the end, and I'd like to introduce our next speaker. Our next speaker is Emily Matilla Valdivieso, and she's in her 3rd year of her PhD. She was also the inaugural president of the QSA and handed the baton over to Harry. She's focusing on using transcriptomics to identify sets of genes that are differentially expressed between tick-resistant and tick-susceptible cattle as part of a biomarker discovery research. She's part of quite a large multidisciplinary project. I'm one of her supervisors, as well as 4 or 5 other people, including Professor Nick Johnson, who started similar research like this in Australia over 10 years ago. So over to you, Emily.
Thanks, Ella. Ticks are the biggest parasitic threat to cattle, causing a loss of $160 million to the Australian cattle industry each year. Treating the infested cattle with acaricides is not a long-term sustainable solution, as ticks are managing to overcome these measures by developing chemical resistance. So my question to the producers that join us here today is, how are you planning to control and prevent the devastating effects of tick outbreaks in light of increasing global temperatures? The good thing is that we have a potentially permanent solution to manage the tick problem, and that is host resistance. Host resistance is an evolutionary mechanism that cattle have developed over thousands of years. We have well-documented evidence that Bos indicus breeds are more resistant to ticks than the Bos taurus breeds. But in crossbred animals, the resistance is widely variable. So far, the only available method to identify this trait consists of counting or scoring ticks on animals while they are in the crush, which is impractical in larger herds. Your time is valuable, so instead of relying on tick scores to check if an animal is worth raising in a tick endemic area, how about using biological predictors of host resistance, also known as biomarkers? This is what my current PhD focuses on. A biomarker is a measurable signature that we can detect in blood and other tissues to predict a particular condition. For example, we have already seen biomarkers being developed and applied in cancer research. But in this project, we are harnessing the power of genomic technologies to capture the information from genes that are switched on and off in animals with high and low host resistance. For the past 2 years, we have worked on this project using Brangus cattle that we identified to be highly resistant and susceptible after experimentally infesting them with ticks for 12 consecutive weeks. We found 2 genes that were consistently expressed differentially between the resistant and susceptible animals throughout the experimental trial. The advantage of these biomarkers is that they can be detected from blood cells and can be used before tick exposure. Future work will involve testing these biomarkers in other independent herds for validation purposes, in the hopes of future industry adoption as a way forward towards achieving sustainable tick control. Thank you.
Thank you, Emily. Right, so let's have a quick look if there's anything in the Q&A. No, there isn't. There is a belated question for Harry for later for Mary to remember. I guess a question I have, and how do you think that these biomarkers, and you may not know the answer and that's fine, do you think they'll be applicable across several different crossbreeds?
We like to think that it will depend on the Bos indicus and Bos taurus content, because if we can associate these genes to the parents, then it will be possible to predict depending on the content in each breed. I think it could be possible, but we are not sure. We will have to validate more herds, more breeds as well.
Yep. Just waiting to see if there's any questions being typed. Ben Hayes has asked, how many genes do you think you need to explain the differences in resistance? And then the question is just 2, so you can answer.
No, actually, this is a very good question, because we know so far that host resistance is a multifactorial trait. And we think that there is several genes, each one of them having little input into what is the final expression of the phenotype. However, this is a good start to kind of like prove or support our hypothesis that biomarkers are potentially useful to predict the phenotype.
Anyone got any more questions for Emily? Don't think anyone's typing. So we might leave it there. Again, there's an opportunity at the end to ask Emily some questions. So well done. Thank you, Emily.
Thank you.
And we'll now move on to Kamran. So Kamran is towards the end of his second year of his PhD. He's primarily supervised by Dr. Peter James, also Dr. Connie Turney, Dr. Ali Raza, and myself. He's looking at factors determining a variation in susceptibility to buffalo flies in cattle. He's trying to develop practical and easy ways to count flies, to phenotype them as resistant towards buffalo flies. He's investigating animal fly avoidance behaviours using accelerometers for animal selection and welfare improvement. And he's also looking at the immune responses of susceptible and resistant cattle using proteomics and immune studies to identify biomarkers that you can use to select for buffalo fly resistant cattle. Also in the spirit of volunteering, Kamran is currently the Centre for Animal Science student rep as well. So welcome, Kamran.
Hello everyone. Buffalo flies are a big problem for a lot of producers in Australia, costing the industry close to $100 million every year. They irritate cattle, cause buffalo fly lesions, and can reduce weight gains by up to 30 kilograms per head over the course of a buffalo fly season. You might have noticed that some cattle have a high number of buffalo flies, while the other cattle in the same herd have only few number of buffalo flies. And we know that these differences are inherited. So my study is investigating how we can measure these differences easily, or easily identify the more resistant cattle so that we can breed for buffalo fly resistance. We can directly count the number of flies, but this is difficult and time consuming. And it's difficult to get good counts because the flies can move between the cattle when they are disturbed. Therefore, I'm looking at different methods to measure fly numbers, such as photography and infrared measures, and looking at different associated factors with resistance in cattle, such as genomic differences and immune response differences. However, today I will talk about an indirect criterion to select animal resistance to buffalo flies, using measurement of behavioral responses to buffalo flies, so that we can identify the more resistant animals. Cattle carrying high buffalo fly burdens show high fly avoidance behavior, such as head tosses, as shown in the image number 2. And we have seen that the amount of these behavior are very well correlated with the number of flies. In particular, the number of head tosses is quite well associated with the buffalo fly number. So, can we measure the buffalo fly awareness behavior as an index of buffalo fly numbers and use this to pick the more resistant cattle? Well, counting the number of fly related behavior would be nearly as difficult as counting the number of flies, and not really practical in a commercial setting. So is there a better method of doing this? As you will know, the use of electronic tags and wearable devices in farm management that monitor cattle behavior that can provide insights for individual cattle performance in an area of rapid development. So could we use these tags to provide an ongoing assessment of buffalo fly numbers? In image number 3, you can see that the ear tags with accelerometer attached that can measure the movements of cattle. So now with the lots of videos of cattle affected with buffalo flies, we are now working with the University of Sydney to develop the algorithms that might allow us to do this. What we are hoping is that by attaching these accelerometers and tags to the animal over the longer period of time, we will be able to electronically identify those animals that have low numbers of flies and use this knowledge to select cattle with the best buffalo fly resistance. This sort of information might also be helpful for doing things such as identifying the high fly number cattle in a herd for selective treatment, or determining the economic threshold for the application of treatments. As I said earlier, we are also looking for differences between resistant and susceptible animals in factors such as immune responses and genomic factors that might be used to pick the more resistant cattle, but that is a story for another day. Thank you for listening.
Thank you, Kamran. We'd like to open that up for questions. Give everyone a little bit of time to type something. So just a question, how will the accelerometer, data assist in phenotyping in the future?
Because with these accelerometers, we are identifying and classifying the different fly avoidance behavior, and these are correlated with the buffalo fly number. So if we are able, because we have seen a good correlation, so we will be using these as an index of buffalo fly number. So as an index of buffalo fly number, we can pick the resistant cattle and use them for breeding purposes in future.
And I think Ben Hayes has got a similar question about whether you've got preliminary results with the ear tags. I'm assuming you mean accelerometers, Ben. Or did you have separate ear tags? Sorry if I missed it.
Yep.
Have you got preliminary results, Kamran?
Yeah, we have visual results and also accelerometer studies as well. And these are actually accelerometer in the picture that are tagged with the ear tags. So we are using them at the moment.
There's another question that's popped in. Have you associated the movement index with blood parameters also associated with fly burdens?
Not with blood parameters at the moment, but with the fly number, yes. Because with a higher number, the behavior responses are higher, and the less number, as you can see in the image number 2, like the buffalo fly number was quite high, and the head tosses are quite high at that time. So at that base, we will be selecting the animals, not with the blood at the moment. The blood study is separate.
So from what I understand from the blood studies that you've collected blood from animals that you've observed already have high versus fly numbers. Yeah.
We have already grouped into high fly carrier and low fly carrier. We are taking blood sample and doing this differential serum proteomics for those. So we can select some biomarkers based on the high and low fly carriers.
Okay, well, we're at the halfway point. This is where I finish off. Thank you, Kamran.
Thank you.
Questions at the end for you. So now I transfer the facilitation over to Professor Mary Fletcher, and Mary will introduce the final 3 BEEF 2021 special speakers. Thanks, everybody.
Okay, thanks so much, Arla, and thanks for organising all of this. Arla actually has to go off and fulfil another role at the university. So it's my pleasure to introduce some more of our students from the Centre of Animal Science. I also work in the Centre of Animal Science, and I'm thrilled to see all the work that's being done by our students. So our next speaker is Chen Chong Ong. She's a QAAFI third-year student. She completed a Bachelor of Biotechnology with honours at the University of Queensland in 2014, and she's now doing her PhD, which focuses on investigating the relationship between cattle reproductive tract microbiome and the cattle reproductive performances. So it's over to Chen.
Hi, good afternoon, everyone. The title of my talk today is a balance that needs to be maintained for better fertility. So during my last trip to the cattle farms, one thing that really caught my attention is that the farmers are very well versed with bacteria. And I say this because when I ask my friends to name one bacteria that they know, most of them would tell me E. coli. While during that conversations that I have with the farmers, they could tell me about like Vibrio, Trichomonas, Brucella, Neospora, and etc. The farmers are so well versed with these bacteria because these are the bacteria which cause infertility and abortion in the cattle farms. And cattle reproductive efficiency is very important for the production in the cattle industry. And I won't go into too much details about that. But today, the main message that I wish to deliver to you is that the cattle reproductive tract is not sterile. Including the vagina and the uterus, they are not sterile. There are millions and millions of microorganisms living inside the cattle womb. And just like our human world, in the microbiome, in the microbial world, there are good guys, bad guys, and some in-between guys. And they all together form this balanced social order within the cattle womb. And everything is fine if this balance is being maintained. However, research has shown that many diseases are actually linked to the loss of this balance in the microbiome. So how did that happen? Like, how do we lose the balance? Let's imagine the cattle reproductive tract as a bank and a robbery case is going to happen. So what does it take for a robbery case to happen? It takes a robbery team, some spies in the bank, security guards who happen to be on lunch break, and a broken security system. So same happens to the cattle reproductive tract. The outsiders, the pathogens, can gain access into the cattle womb during mating or calving. And this is actually fine if the good microbes and the host defense system are in place. But what if the good crops were distracted and the security system was down? This is when the nasty thing will happen. And we often think a disease is caused by one particular microbe, one particular pathogen, and we even name the disease after the bacteria. But if you realize, I have been saying a robbery team. Yes, it takes a team. Like in a robbery team itself, we have like someone who is in charge of the escape plan, someone who breaks the security system, someone who distracts the good cops, and someone who is actually doing the actual deal, which is getting the money. So during cattle reproductive disease development, the same things happen. Fusarium bacteria is in charge of weakening the host defense system. Prevotella is in charge of tearing down the uterine wall, while other microbes can do the actual thing, which is like directly infect the sperms, the eggs, or even the developing embryo, and eventually leads to infertility and abortion. The good news is that the cattle reproductive tract microbiome can change, and in fact, it has been changing just by nature. The same cow can have different microbiome in the cattle womb during different stages of reproductive cycle, during different stages of pregnancy. So what QAAFI is doing now, which is also my PhD project, is to identify the healthy and balanced microbiome in the cattle reproductive tract, and also to identify the differences between a disturbed microbiome and a healthy one. So we wish to come out something we call microbiome index, which serves like a ruler to measure how distorted this microbiome is from the balanced one. So what are the potential applications of this microbiome index? Because we know when the robbery team will come in, either during mating or calving. So what if we can use this microbiome index to boost up the good crops, the good microbes to the right level, to help them to maintain the balanced, healthy microbiome even when the intruders come in? And I'm also very looking forward to that day when we can use this microbiome index to restore the balance in the cattle reproductive tract microbiome and consequently restore his or her fertility, even after something goes wrong. You know, give them a second chance. That's all from me. Thank you.
Thank you, Chen. That's a very good analogy with a robbery bank. Although we did have a question from Jeffrey about how do you know so much about robbing banks? But that's Jeffrey for you. I will leave you to have
that Jeffrey, that happens a lot in the city.
Okay, so I would leave it to have some more serious questions online there. From me, is there much difference? You're talking about organisms getting in, the difference in older animals or animals in different areas? Are we expecting different resistance?
Yeah, so same compared to like the previous topic we talked about. Cattle reproductive efficiency is also multifactorial. So we will have to do like a huge data in order to come up with something that it's that consider all the other factors. For example, their age, their health, their nutrients level, and the environment that they are living in. Yeah.
Yep. Okay, so you've got a couple of questions. One from Ben Hayes, who says, fantastic job. And how many species are in the reproductive microbiome?
From the published papers and from the pilot studies that I have done so far, in terms of species, they can go up to like few 10,000s.
Oh.
Yeah, in terms of species, yeah.
Mhm.
But when we look at just the phyla, we have the 5 main phyla.
Okay, thank you. So the last question on there is, how do you envisage to use this research in a practical way?
Use this research in a practical way.
Like how would you help
translate
the
to the
farmer
field?
with it?
Yeah. I guess this is cattle reproductive tract microbiome is still like very new. In fact, the whole microbiome research field is still very new. But we have seen some examples of how people are using like the microbiome in guts, like how they give the probiotic and stuff. So what I was talking about the microbiome index, if you can come up with like to predict like what is better for fertility, we can also inject like bacteria into the right parts to help with that.
Okay, thanks, Chen. That's a very informative talk and very well explained. So we're on to our next speaker, who is Noman Naseem. Noman's also a third-year PhD student, but he's quite a different background. He's got a veterinary degree from Pakistan, and he completed his Masters of Philosophy in Veterinary Pathology before he started at QAAFI in 2018. So his PhD investigates the pathology and pathogenesis of buffalo fly lesions. So over to Noman.
Thank you, Mary. Hi, everyone. The buffalo fly is a small blood-feeding ectoparasite that feeds mainly on cattle. In Australia, skin lesions in cattle associated with buffalo fly feeding are termed as buffalo fly lesion. These lesions occur most frequently near the corner of the eye, neck, or at the dewlap or on the belly, and can range from raised, dry, hairless, scab-encrusted areas to open bloody wounds. These lesions have significant impact on cattle welfare and can cause the cattle to be rejected from export or bring lower prices in the sale yards. However, the exact cause of these lesions is still not clear, and there are no methods to control these lesions except controlling the buffalo flies. So to develop a specific control method for these lesions, it is necessary to rule out its pathogenesis first. So my project is investigating the pathogenesis of buffalo fly lesions to identify the factor involved in its causation. So here is the first culprit, buffalo flies. There appears to be a poor relationship between the buffalo fly number and the lesion development. I mean, the fly carry high number of, cattle carry high number of flies did not develop lesion, while the cattle with lowest number of flies develop severe lesions. This indicates that there is something else involved in the lesion development. Buffalo fly was reported to transmit a small nematode belonged to genus Staphylophleria, which was speculatively implicated in the pathology of lesion. Although whether these nematodes play a key role in the development of lesion or just occur opportunistically is not clear. There was no specific molecular detection assay reported for this nematode previously, and no genome sequence information was available for this genus in sequence databases. So to clarify the role of Staphylophleria in the lesion development, accurate detection of this nematode in the flies and the lesion was necessary. So that was my first challenge, to develop a highly sensitive and specific detection assay to accurately detect that nematode. So I have isolated the nematode from northern Australian cattle and identified it as Staphylophleria by scanning electron microscopy. And then I developed a qPCR based assay. Our assay was specific and highly sensitive. With these novel assays, I have identified a nematode in the buffalo flies and lesions from northern and southern and central Queensland, but I could not find any sample positive from South Queensland. But lesions are still here. That means there is something else. And that pointer lead us to check for the potential bacterial involvement in the lesion causation, because there was no previous study on the bacteriology of these lesions. So I have also isolated Staphylococcus hycus, both from lesion and the flies. And interestingly, that finding is common between northern and southern cattle. But how this bacteria involved in the lesion development is still not clear. So I'm also looking for the potential virulence factor in the isolates through whole genome sequencing. In the last, in a herd, not all the cattle develop lesions. Some cattle are naturally resistant to the lesion development and some are susceptible. And it is generally recommended that to get rid of these lesions, cull the allergic cattle. But exactly what does that mean is not now, like it is allergic to buffalo fly or nematode. So this project further aimed to check the role of hypersensitivity or allergy in the lesion development, and also determine the differences in the immune response between susceptible and resistant cattle. In one line, my project is investigating the pathogenesis of buffalo fly lesion, and the comprehensive knowledge of these mentioned factors will help us to design specific control strategies for these lesions. Thank you.
Thank you, Norman. Very interesting. I was interested in the fact that your nematodes, you found them in the north, but not in the southern regions. Is that been observed in other parts of the world?
No, that nematode was already reported from different parts of the world as a cause of cattle skin lesion. And but in Australia, it is not very much consistent in the lesion. And I'm also doing the histology, and I have seen that the presence of nematode just worse, or that just make the lesion more worse. And it is not like these nematodes are not necessary to start the lesions.
So it's like a stepwise process, the bacteria
Yes.
first,
Yeah,
followed by then the nematodes
yeah, it might
are
be
later.
might be multifactorial pathogenesis. So.
Yep. Okay, I can't see any other questions at the moment. Oh, no. Jeffrey's.
Yes, Jeff.
Jeffrey's asking a similar questions. Are fly lesions an issue in other countries?
Jeffrey, hornfly, which is another homologue of like buffalo fly, it also caused the same lesion in US cattle, and they found that they also have the stiffenofilaria, they also have Staphylococcus aureus, and they also reported lesion due to hypersensitivity. So in their cattle, it is due to like they have a multifactorial pathogenesis, but they didn't reported any stiffenofilaria from last 20 or 30 years. So.
Okay. I'm just keeping an eye on the time, Norman. We're actually tracking really well. So I'll leave that. If there's any other questions for you, they can ask them at the end. So we're moving on to our sixth and final speaker, who is James Copley. So James is a second-year PhD student. He actually did a bachelor's degree with honours in ag science in animal science, specialising in animal science at UQ at Gatton. So a different background again. And he commenced his PhD in 2019. And he's investigating the genotype by environment interactions for beef cattle fertility traits in Northern Australia. So over to James.
Thanks, Mary. Afternoon, everyone. I'm James, and I'm here to tell you that fertility rates in Northern Australia are trash. Fertility is the ability of breeding females to produce a calf annually, and is suppressed in Northern Australia for a variety of factors, but largely due to the harsh environment. In the north, the challenge has been and remains to get heifers to cycle, that is, reach puberty prior to joining, usually for 2 years in the northern context, for them to conceive for the first time and calve at 3, and it is very challenging to get them to rebreed for the first time, because heifers in this situation are growing and lactating at the same time. So what is the scale of the fertility problem? In the ongoing Northern Genomics project, which is MLA funded, 3 fertility phenotypes have been recorded for 25,000 heifers from 54 commercial herds from diverse environments across the north, which you can see on the map on the slide. It was found that less than 50% of heifers were pubertal at 600 days of age prior to joining. 72% of heifers were pregnant at first pregnancy, but only 63% of recorded heifers were successfully pregnant at second pregnancy. And these results are much lower than the anecdotal evidence that you might hear around the traps at Beef Week. Fertility might not be a problem for everyone, but improving fertility is a challenge for the industry as a whole. Increasing reproductive rate by a single standard deviation will result in a greater improvement in profitability for beef businesses than chasing weight gain, which is a bit of an industry obsession. The harshness of environment generates a catch-22. The harsh environment requires cattle with a high Bos indicus, that is tropical cattle content, but this has a penalty for fertility, as Bos indicus cattle are notoriously older and heavier at puberty. So my key question is it possible or better to select for adaptation in the more fertile Bos taurus cattle, or is it better to select for improved fertility in the already highly adapted Bos indicus cattle? Hence, my project is looking at genotype by environment interaction for beef cattle fertility traits. G by E is the concept of different genotypes being affected differently by environmental variation. If I have an Angus and Brahman genotype side by side in a southern environment, there's little debate that the Angus will outperform the Brahman genotype in terms of fertility and production. But these same two genotypes side by side in a harsher northern environment will both see a reduction in productivity, but the Brahman will reduce less. The genotypes associated with maintaining fertility despite harsh environmental conditions are the most valuable in Northern Australia, and it's those that I'm seeking to identify. My project is being conducted in 3 phases. Firstly, I want to define and have defined environmental descriptors relevant to the challenges faced in Northern Australia. Firstly, the availability of nutrition, for which I've used weight gain information and daily rainfall records for each of the locations present in the Northern Genomics dataset. And secondly, exposure to heat stress. I've used daily temperature and humidity information to define when animals were exposed to severe heat stress throughout the breeding cycle. The second step is to verify the presence of a significant relationship between the environmental descriptors and the fertility phenotypes from the Northern Genomics project. For example, I'm hypothesizing that exposure to severe heat stress will reduce the probability of pregnancy at pregnancy 1. My third step, which I'm currently working on, is to incorporate the genomic data into the analysis. This will allow me to specifically identify the different response of genotypes to environmental variation. My results so far, principally I found that heifers that were pubertal at 600 days of age had an increased chance of successful pregnancy at first pregnancy, underscoring the importance of early maturing cattle that are pubertal at joining. I found that increased average daily weight gain in the period leading up to pregnancy 1 and pregnancy 2 significantly increased the rate of successful pregnancy. That's not a surprise, but it does verify the use of a nutritional descriptor in this analysis. And the results suggested that heat stress had a significant and negative impact on reconception for success at pregnancy 2. But it did not have such a significant impact on attainment of puberty and success at pregnancy 1, suggesting that heifers that are lactating, in the case of those at pregnancy 2, are more susceptible to severe heat stress. The aim of my project is to select for cattle that can perform well for fertility despite harsh environmental conditions, and thus improve fertility and profitability across the industry.
Okay, thank you, James. I'm just waiting to see if we got some questions, but nothing as yet. You're talking about doing the G by E for the northern regions. Does it equally apply that you'll have other factors in the southern, more fertile pastures?
Other factors, Mary?
Sorry, other different outcomes, I guess. Sorry.
There are certainly different fertility outcomes. Is that what you mean?
Yeah, well, okay, your study is more focused on the north, but
yeah, well, I mean, it's a range of environments. Some of the environments that are represented in this data set are quite favourable.
Okay.
So there's northern and northern, isn't there? So I think there's a full spectrum present in this data set. Some of the environments here are just as favourable, if not more, than quite southern environments.
Yeah. Okay. Sorry, we're not seeing any questions. If any of the audience have... Okay, well, we might wrap that up and open it up to the panel again. And if there's any more questions for James, he can answer those as part of that. So there were a couple of unanswered questions going backwards. There were a couple from Robin Warner for Harry. So if Harry's still online? Yep. So Robin's questions, she had two actually, is about your work, is how long do you think until it would be available? Obviously wanting to have that thing, the detection crash side. So what sort of timeline do you think you're looking at? So, sorry, Harry, we can't hear you. Don't understand why.
Sorry, Mary, we've just had to change the laptop here.
Technical difficulties. We've done well so far.
Yeah, so we're hoping to sort of within 2 years to be able to trial the pipeline, and we'd be trialing this on sort of collaborating properties and stuff like that, and then we've got to look at really making it possible to run it. For producers to run it, but also making it economically feasible. And that's really the big thing. There would be a premium paid in the northern beef industry to be able to genotype your animals on farm, because the northern beef industry really doesn't lend itself to being able to send a sample off to a lab and then wait until you get the results back.
Mm-hmm. Okay, Robin's second question was about breeding values. So will this help us develop ASBVs for the beef industry similar to the sheep industry?
Yeah, so a lot of southern producers already will be using genomic estimated breeding values in their beef herds, but the northern industry just hasn't been able to adopt this because of the limitations around sending samples off to a lab. So, for example, a property I worked on only had a male run every 2 weeks. So it's very difficult to send your results off, wait 3 months to get your results back. So that's been the limitation for them adopting this sort of genomic selection.
Okay, I think there was a question from Jeff that we didn't get to for Emily. So Emily, do you have any evidence for the specific action of the two magic genes? Are they associated with susceptibility or resistance?
Thanks, Jeffrey, for the question. So these two genes, the magic genes, turns out they are involved in actually inflammation and the recruitment of immune cells, which makes sense because this is what differentiates resistant and susceptible animals. Usually we think that resistant animals are better at handling these responses, and that's why we see these two genes being downregulated in these animals. Yeah.
Okay. Oh, look, there's a question for James, come on, from Ben. So if the Earth warms by 2 degrees, how will pregnancy rates be affected? Can you calculate this from your data?
I can certainly estimate how big effect that's going to have from my data, because I have a good spectrum of temperature versus pregnancy information. So the modelling so far suggests that that it would be reduced. An increase in global temperatures would reduce the pregnancy rate.
Okay, look, I think we've got one final question for Harry about, if he's still there. Okay, Harry, multiplex animals per cell per run, how many? That's from Tim.
Yeah, cheers, Tim. So we're sort of working around the magic number of being able to get 20 gigabytes of data off each flow cell consistently. So 20 gigs represents sort of 6-fold genome coverage. But then when we go and incorporate techniques like selective enrichment, where we can actually enrich specific loci when we're sequencing while depleting non-informative regions, we're hoping to hit sort of 10 to 20 animals comfortably, and then go up even further once we incorporate stuff like very low coverage imputation pipelines. We think we could get upwards of sort of 40 animals.
Okay. Look, I'm super impressed that everyone's kept so well to time. And also all the fantastic science that's been covered here. So I think that just about wraps up our science seminar for this week.